Hello, everyone. My name is Sylvia. I'm the Product Marketing Manager at Medidata. Thank you for joining Medidata's live webinar, Optimize and Transform Clinical Oncology Research today. The webinar will last for an hour. At the end of the webinar, we'll have around 10 minutes time for a Q&A session for the audience. Feel free to write any questions to the chat box, and we will collect all the questions for the speakers to answer during the Q&A section. Before we start the webinar, I would also like to introduce our team members who are here to answer some of the questions today. Jasmine is our Regional Account Manager. Cheetah is the Solutions Consultant. Arenas is the Market Development Specialist, and also myself. So for now, please welcome our first speaker, Mark Maclean, Head of Clinical Data Management at the NHMRC Clinical Trials Centre at Sydney University. Mark is responsible for CTC's clinical data collection and management systems, and overall standards for the clinical data lifecycle. Mark has a background in radiation oncology and clinical research, and over 20 years experience in systems development, implementation, and training. He will be talking about the trends and challenges from the academic research organization perspective towards precision oncology. So Mark, over to you, please.
Thank you, Sylvia, and thank you everybody for the opportunity to talk to you today. As Sylvia said, I work at the Clinical Trials Centre as part of Sydney University and head up the clinical data management group there. So what I'm going to talk about today, I'll just give you a brief overview. I don't seem to have control of the, there we go. So I'm going to start off with a very brief background on the CTC to set some context, the way that we see academic trials against industry trials, some of the key differentiators, and then I'll talk through in a bit of detail about the kind of work that's going on in precision oncology around Australia, New Zealand, and then across the world, and the trends that we see, the new developments that are coming up that are leading to some of the challenges that we have in collecting and managing clinical data for investigator-initiated studies. So to start off, the NHMRC Clinical Trials Centre has been established 30 years this year as a centre of excellence, part of the University of Sydney for clinical trials. We now have more than 200 staff, a multidisciplinary team with a number of specialisms spreading across clinical trials core team, statistics and research methodology, translational research, health economics, labs, systematic reviews, and health technology assessment. So we have a wide spread of expertise in the centre, and we work all in combination for the studies that we develop. Over the 30 years, as you can see, we've had more than 100 trials and more than 70,000 patients on trials that we've run from the CTC. And the way that we work is we develop the whole study, work with investigators and trial networks, clinical groups to develop the study, develop the protocol, manage all of the ethics and regulatory submissions, act as sponsor, and then execute the study, analyze and publish. So it's what the Americans would call, I think, soup to nuts. We do the whole study, run and manage that at the Clinical Trials Centre. We work with a network of different clinical research groups, access to over 800 investigators around the world, and we're typically running 40 to 50 active concurrent trials at any one time. The therapeutic areas that we cover are oncology, which is the majority of our studies, cardiovascular disease, diabetes and obesity, neonatal and perinatal work, primarily around premature babies, around improving outcomes for very premature babies. In oncology, we work we focus on solid tumours and work very closely with collaborative groups of researcher clinicians in the areas of gastrointestinal tumours, gynecological, urinogenital and prostate, neurological, so brain tumours, lung, including mesothelioma, and then a number of subspecialties as well. We also have a great interest at the Trial Centre in patient-focused research. So just about all of our oncology studies, unless they're early phase 2, will have outcomes related to quality of life impacts on the patient and even patient preferences. So how would we characterize investigator-initiated studies as distinct from those for compound and drug development? The defining characteristic, I guess, is independence of the design, the conduct, and the analysis and publication of the study. We work not for commercial ends, and it's really the focus of our work is producing evidence to increase the evidence pool available to advance and optimize existing therapies. We often run studies that are combination therapies, for example, agents from different biotech companies, or quite commonly also multimodality, so using particular agents in combination with surgery or radiotherapy, maybe a study to check whether pre-adjuvant or adjuvant chemotherapy is better in a certain surgical context. Our funding is limited compared with industry studies. We would say that we have limited funding from our perspective. The majority of our funding comes from government grants from the National Health and Medical Research Council, which is across Australia, or from charities. Another big funder is the Cancer Australia and other cancer institute bodies that provide grants for investigator-initiated studies. But we have the same challenges of ethics and governance and approvals as anyone else running clinical trials, so we have to play by the same rules. Compared with some other things I've read recently about how the world sees investigator-initiated trials as an academic research organisation, I think we're a little bit ahead of a single site or couple of site pilot studies that may be run by an investigator. We do still use some paper CRFs on occasion, but normally just for pilot studies to test out the feasibility or to further refine the data collection requirements. The trouble with investigator-initiated trials is that you tend to end up with a multiplicity of data systems, more data systems than you'd care to think about, and we're working very hard to smooth that out and rationalize things so that we can get all those efficiencies. The other thing about working with a panel of investigators is everybody's got their own idea and everybody's got their own preference, so that can be a challenging discussion sometime. Over the years, we have significantly tightened up on the operational data management, the data cleaning of studies, and in particular, we would say we take a very pragmatic and risk-based approach to on-site monitoring and data cleaning, data management. We have a lot of central monitoring, risk assessment and risk management of our studies. We would say that we've been doing risk-based monitoring for many years, although not with the science and the facilities and the statistical support that is now common in industry. We would do limited source data verification, focusing on the key data points. So almost without exception, we've never done 100% SDV. Pragmatic is the key word. Okay, so that's probably covered that. Let me move on now to talk about, oh, sorry, I'd forgotten about this slide. This is an excerpt from a report by the Australian New Zealand Clinical Trials Register, which is the equivalent of clinicaltrials.gov for studies running in Australia and New Zealand. And you can see it gives an overview of the characteristics of studies over the last 10 years. This is the most recent report published in 2017, but data up to 2015. And it shows, I think, quite different to clinicaltrials.gov, that 63% of studies run across Australia had a non-commercial sponsor, and about 45% of the studies had no industry involvement. Now, if you take those as proxies for investigator-initiated studies, then that's the majority of study work that is going on that isn't outside of the industry domain, which I think is quite interesting. Okay, so what is happening moving towards precision oncology? What is today's oncology investigator-initiated trial actually looking like? Of course, there are phase 1 units. There are many phase 1 units around the country, but particularly from the perspective of an academic research organisation such as ours, a typical study is phase 2 or phase 3, may or may not be randomised, and would typically have multiple stratification factors and or multiple cohorts of patients, so pretty complex before we even start talking about newer developments. Quite a few of our studies also are designed as a phase 2-3 protocol, and that's to get around funding restrictions. So we'll write the protocol as if it were a phase 3, but specifically design it with analyses in order to say, well, we've currently got funding for a phase 2 study, let's do that, analyse it, and then if the results are on track, then we'll move on to the phase 3 subject to further funding. A typical oncology study in our space would have multiple objectives and endpoints built into the protocol, and a combination of those 4 aspects there. The study would run for 2 to 3 years recruitment, 3 to 5 year follow-up, and sometimes first-line treatment, but more often second or third-line treatment for cancer patients. So the total duration of a study that we run, it's really unusual for an oncology study to run for a duration of less than 5 years in our work. A lot of the hard endpoints would be imaging-based, so we have a high requirement to collect the imaging data, so we have modules to collect and clean RECIST, iRECIST, and RANO data very commonly. RECIST, I'm sure most of you are familiar with, response evaluation criteria in solid tumours. And in the last year or 18 months, we've been doing a lot of work with iRECIST, which is immune, it's a modified RECIST taking account of the different response criteria that you can get when treating cancer patients with immunotherapy. In essence, the difference between the two is measurements are taken in the same way, but the assessment of response is different because certain classes of immunotherapies, the UMABs, for example, have been shown to occasionally cause a pseudo-progression in the tumour as the immune storm takes hold. The tumour can appear to swell up, which could be seen as progression, but in fact is just temporary. It increases in size and then, if all's going well, rapidly shrinks. And also you can see new lesions appearing where in fact they were present on the baseline scan but were too small to discern. But if you get a strong immune response, they swell up, looks like a new lesion, and then next scan it's gone. So iRECIST was defined, I think, primarily by a group led in Canada, the Canadian Clinical Trials Group, with Leslie Seymour as one of the key authors of that paper, and then we were working alongside her and ran some of the first studies measuring iRECIST in solid tumours. Another common theme in today's oncology is tumour mutation profiling, genetic signatures, protein signatures for both eligibility and stratification of patients in our studies. And that is now very common where we're working, of course, with certain drug categories or biological categories. So as we think about precision medicine, something that one of my colleagues mentioned to me is that precision medicine, as well as targeting tumours and getting really good response for subsets of patients, for scarce types of patients, can also help you not do something. An example would be by finding no additional benefit of a new agent or a combination of treatments, you can actually save the patient's morbidity. An example being there was a study in breast cancer looking at low risk, medium risk and high risk patients, and the study determined that for the medium-risk patients, is it worth combining chemotherapy and hormone therapy? And the precision study said no, there's no additional benefits, so we can spare that large cohort of patients unnecessary treatment. So that's how we see today's study. What are the trends in investigator-initiated trials in oncology from our perspective, from an ARO perspective? Laid out on the slide there, I'm going to talk in more detail about a couple of them, but the first, and to us the most obvious thing, is the increasing complexity of study design, the increasing degree of change of study designs, and the need to adapt the study that's running for 4, 5, 6 years as new information comes available, either on the study or from other research published at ASCO and similar conferences. Tumor genetics and looking for mutations, actual or potential biomarkers, is a massive growth in our studies. And there has been a massive growth in translation the last couple of years, and I'll talk more about that in a moment. Immunotherapies, as I've said a couple of times, are showing great benefits for subgroups of patients, for selected cancer types, and the knowledge about them is evolving month by month as new work is undertaken and published, and we try our very best to keep pace with that and adapt our studies and our protocol designs as that information comes out. There's, alongside that, there's drug repurposing. Now, that is common in the academic world. I don't know how much it would happen outside the academic world, where you're looking at an existing, well-established, often a very old drug to see if it's got any additive or adjuvant benefits in combination with newer therapies. 3 examples of those for studies that we've run at the CTC. There was a trial called MATES, M-A-T-E-S, in malignant mesothelioma, which was using maintenance thalidomide in combination with other agents to maintain non-progression status in those patients. We did a study in early bladder cancer, which looked at a combination of mitomycin C and the BCG vaccine delivered intravesically into the bladder to, again, prevent progression in very early stage bladder cancer where it hadn't invaded to the muscularity of the bladder. And we have a study called SPAR that's opened in the last month or so, which is using simvastatin, a very old established statin drug, to induce a greater, it's blinded placebo-controlled. The hypothesis is that it would induce a greater tumor regression with pre-surgical chemo and chemoradiotherapy. So drug repurposing is definitely a trend that we try to follow and support. And there's a move away from organ-based interventions in the master protocol design and the translational research, which I'll talk through more in a little while. So, translational research, the way we use the term is primarily the discovery and the research into potential biomarkers and patient selection rather than translating the evidence into practice, the other aspect of translational. TI is now embedded in basically all of our phase 2 and 3 oncology studies. It's used for eligibility checking and cohort selection. Obviously, if you're checking eligibility based on a certain genetic profile or mutation, that can cut down your available patients, but the theory being that it would give you a far more targeted, far more precise intervention study. The 3 examples I've got there are studies that we've run. 2 of them are in brain cancer. Virtue used genetic profiling and also looking at the methylation status of the MGMT in glioblastoma multiforme, GBM. They're very aggressive brain tumor. Nutmeg is also looking at that tumor type, but with a different patient population, older patients, and that is using nutmeg to screen and stratify the patients into, in effect, high and low risk patients. And Embrace is a basket study, which I'll talk more about in a moment, which is looking at both breast and ovarian cancer, screening for germline mutations in the BRCA genes. So all of those bring a big lot of, a huge amount of data when you're looking at the omics, and we need to have mechanisms to link that to the clinical data that's collected on the study, of course, and we're generating tens or hundreds of thousands of biosamples and data points along the way as we embed translational research into all of our studies, really, we would say at the leading edge. We also have focused translational research studies. One of them is called AutoCheck, where we're reusing samples and data prospectively from ongoing studies in immunotherapy to look at the molecular determinants of autoimmunity and immune-related adverse events. That is one of the studies that evolved and changed as the immunotherapy and iResist was coming out. So it's quite challenging to connect one study with 6 others that are already running and already changing, a lot of complexity. We're also doing liquid biopsies, looking at circulating tumor cells and DNA. But as I said, that whole genomics, proteomics, anything omics generates a lot of data that has to be managed. The other aspect of our translational research is we're seeing far extended data life cycles. So with the CO17 is a study that closed to recruitment, I think more than 10 years ago. We ran the Asia-Pacific component of that for the Canadian Cancer Clinical Trials Group, and we are now actively using the biosamples that were taken on that study and all of the data associated with them and the clinical data for new active research. So it's significantly extended data usage times. The other aspect, which I'm sure most of you will be familiar with, another major trend, is the use of master protocol study designs. So they are, if you haven't come across them much, it's the idea of having a single master protocol in which you design it to allow diseases or treatments to be added or removed from the study, and you can have, in effect, sub-studies or sister studies evaluated concurrently or sequentially in a far more efficient way than running multiple separate phase 2 studies. In order to support that design, you need to have centralized systems that are capable of dealing with a lot of change to add and remove treatments, change cohorts, add cohorts, and so on. And you definitely need to work to get a set of common CRFs that can be leveraged across all of the related studies under the master protocol. Agreeing a set of standards in the academic world is a challenging and joyful experience, and we're working very hard to firm up on that. The types of master protocols can be characterized as umbrella, basket, and platform. I'll talk a bit more about those in just a moment. The terms amused me when I first came across them, but they're now standard in the investigator-initiated oncology world. So, umbrellas and baskets. The idea of an umbrella study is the type of platform study where you're looking at multiple targets within a single primary type. So multiple genetic targets or susceptibilities of the tumor, attacking those with multiple drugs based on a single primary type, for example, ovarian cancer. And the idea is you've got a single disease, looking at the targets, you approach it with different targeted therapies, and they become different cohorts or sub-studies within the platform. Looking at it the other way up is a basket study where you have a variety of different disease types, different organs of origin of the cancer, and you exploit different histologic or genetic features. You target those with different drugs.
So kind of the opposite, where you can have multiple organs all targeted with a common therapy. Another way of looking at these are what are called single signal seeking studies. We're running a program at the moment called MOST, the Molecular Screening and Therapeutics Program, which is a framework protocol for multiple parallel studies looking for signals of activity of novel molecular targeted agents for patients with advanced cancer. So typically, we're looking at third or fourth line treatment where patients have exhausted all standard options. The patients are derived from registers or biobanks sometimes. There's a very complex screening and selection process, and then rapid treatment initiation. So you need lots of efficiencies of design and operations in order to realize that paradigm. And the last type is adaptive platform designs, where the study is designed with pre-specified planned flexibility to add and drop treatments for a particular disease target, and you can add or drop. You can drop treatments for futility or declare one treatment superior to others, and then rapidly move on to another cohort of patients or to move into a phase 3. So an example of that, as I just mentioned, is the MOST study. You need the infrastructure to support that, to have the flexibility, and another example of them is the couple of studies that are running out of the US. I-SPY2 was a very innovative study using Bayesian adaptive randomization methods. Don Berry and SciAps were key in that activity, and they're also working on GBM Agile, which is a global adaptive platform for GBM, for glioblastoma tumors, in what they're calling an interactive learning environment, where it takes real-time feedback on patient progress and progression in the study, and then feeds that back to adapt the randomization model. Very innovative stuff. So to conclude, where do we see the challenges in precision oncology? What are the things that are stretching current capabilities and real opportunities to move forward? Laid out on that slide, it all, in my mind, adds up to increasing complexity. You have massive growth in data volume, data sources, and a need for bioinformatics, a need to adapt studies in flight as new information becomes available, or you get real-time feedback from the study. Omics data, translational research, huge amounts of data that needs to link back to the clinical data. We're managing tens and hundreds of thousands of biospecimens in virtual biobanks. The challenges around that include managing all of that data, having a data governance structure, having a mechanism to link and reuse all of that information. We have a lot of specific eligibility, and the knowledge is changing all the time, as I've outlined a couple of times for the immunotherapy examples. The final aspect that I've drawn out there is that you have global teams and global data. It's no longer restricted just to the one sponsor in the one location. For some of our brain cancer studies, for example, we're working with collaborators in the US, even though the patients all are in Australia or New Zealand, working with collaborators in the US for very specialized imaging-based adjudication and imaging review. So you need a system that allows you to share that data in an efficient way. So that's all I was going to say today. The finishing thought that I've got is that as we increase precision, we massively increase complexity and data volumes and the need to link it together. So you need a platform that's stable and adaptable to take that forward. And if any of you have got kids, you'll probably recognize that's from Minecraft. And it's that sort of flexibility that we need in our data systems these days. Okay, thank you, Sylvia.
Thank you, Mark, for the wonderful presentation. Now, we will pass the time to our next speaker, Michel Becci, Vice President of Industry Strategy at Medidata Solutions. Michel has nearly 30 years of experience in the life science industry. Since joining Medidata in 2013, Michel is responsible for helping to shape the industry strategy of the Medidata portfolio. She holds a bachelor's degree in microbiology and an MBA in international business. Now, Michel will talk to us about how to address the challenges Mark mentioned about and how to optimize and transform clinical oncology research. So over to you, Michel, please.
Thank you so much, and good morning, everyone. Thanks for taking the time and a few minutes with us today. So I wanted to spend the next 20 minutes or so walking through how we at Medidata believe technology can really help address the challenges that Mark has identified. So firstly, thinking about innovation in the space, and Mark spoke to this, but you know, I've been in this industry for close to 3 decades now, and it's really, to me, exciting to see the pace of innovation in this space, and especially in oncology research. The pipeline in oncology is growing substantially, as we all know. The number of active compounds in oncology R&D has actually quadrupled since 1996, and it's nearly doubled since 2008 alone. So given this new wave of cell and gene therapies, these new frontiers being explored, these changes in the clinical development paradigm, such as Mark mentioned, I think with all of this innovation, we also tend to see the challenges that Mark laid out. And as I think about in Mark talking about the complexity of the trial paradigm today, I think I would bucket these challenges into 3 areas: patient scarcity, clinical complexity, and data abundance. So firstly, let's look at each of these individually. And first, looking at patient scarcity, first just a couple of stats, and I'm sure you're familiar with some of these, but 50% of trial sites fail to meet enrollment goals today. 70% of patients live more than 2 hours away from clinical trial sites, and there are greater than 1,000 immunotherapy trials today that require a targeted patient pool. So this patient scarcity challenge is real, and how do we, given the aggressive timelines in clinical research, overcome the patient scarcity challenge? And so we think about it in 3 ways. 1, reducing the patient burden to improve not only retention, but actually recruitment as well. How do we make it easier for these patients to actually participate in trials? And then how do we utilize tools that will help find those right patients for the right trials? So let's look at each of these areas independently. So first, reducing the patient burden. At Medidata, when we think about the clinical trial process, if you will, the first step for us is really around efficient study planning. And it's critical that you design protocols with an eye to both the patient and the site burden. We have a tool called Design Optimizer, which looks at protocol complexity as well as patient burden, and gives you the capability to take a look and benchmark the procedures that have been selected for that protocol against other procedures in a similar therapeutic area. Additionally, we've recently added something called the Patient Burden Index, which is an analytical metric that utilizes the actual patient schedule of assessments to determine the hardship assumed by the clinical trial patient. So by keeping an eye of the burden to both the patient and the site, we believe that this is one of the ways that we can improve both patient recruitment and retention throughout the trial. Secondly, making it easier for patients to participate. As we all know, the process of being in a clinical trial for patients can be really confusing. Patients are presented with a ton of information. The protocol can be very confusing. Informed consent documents can go upwards of 30 to 40 pages. And then once those patients are consented, they then have the burden of coming in for follow-up visits. And as I mentioned, given that trial patients can live so many hours away, this can be a very difficult endeavor for them. So the easier we can make it for the patients, the better chance we stand of enrolling them and keeping in the trial. So at Medidata, we focus a lot on the patient journey through the platform and trying to help simplify this process from start to finish. So by having this unified platform, firstly, we think about educating the patient on the protocol electronically, and then secondly, the ability to electronically capture the patient consent to really ease that burden for the patient right from the start of enrollment on the trial. And then as the trial progresses, it's all about having the digital capabilities, whether from things like ePro or eToa, or collect data from apps and sensors to conduct the study so that the patient experience is improved throughout the process, keeping that patient experience paramount throughout the process. And then lastly, with all of this additional digital data coming into the process, it's also critical that you're seamlessly able to sync and analyze that data with the other clinical trial data in the process. So for us, it is thinking about this patient journey from the start to the finish of the clinical trial to really make it easier for the patients to participate. There's a stat saying that almost 80% of cancer patients are diagnosed in a community setting and therefore require logical support to really reach the nearest trial center. So obviously, the better it is for the patient to remain at home, minimize disruption in their personal lives, we believe that we'll be able to really improve enrollment in a trial. So we talk a lot at Medidata about something we call the trial dial, which enables in the same platform to have virtual trials along with site-based trials. And depending on whether the study requires 100% site-based, 100% virtual, or something in between, you're able to use the same platform to spin the dial, if you will, and use the appropriate amount of virtual visits along with the realities of having the patient actually having to come in for in-person tests as well. So although these trials are far from mainstream today, we do believe that they'll continue to increase. And a good example of such a study is the adaptable study that Medidata has been involved in. This is it's in year 2. It's the largest virtual trial ever with a target of about 15,000 patients to enroll. Those patients are actually being enrolled through electronic health records, and to date, the trial has recruited about 7,000 patients with another 8,000 to go. And the study is actually looking at aspirin dosage levels on patients with heart disease. Again, one of the largest virtual trials ever. We're very proud to be part of this study. And again, although these studies are far from mainstream, we do believe that as we can continue to improve the paradigm and improve the burden to the patient, that will really help the patient remain at home and improve the way that clinical research is done. Moving on to utilizing tools to help find the right sites and the right patients for the trials. At Medidata, we do have a study feasibility tool which does enable sponsors and sites to actually identify sites, have site recommendations based on that site's industry experience, actual key performance indicators in previous studies, and also advanced analytics. And through this, it offers an approach to not only understand the performance at various study sites, but also sponsors can learn from prior successes and failures, but then also understand where there's a higher probability for identifying and recruiting relevant patients at specific sites. In addition to that, Mark actually mentioned SciAps earlier in connection with one of the trials that he's been involved with. But we've announced a partnership with SciAps probably about 6 months ago now. You can find a lot of additional information about the partnership on our website. You'll be hearing a lot more about our partnership with SciAps. But we are jointly looking at a way to develop solutions to make it easier for oncology patients to identify trials. And so leveraging our combined partnerships with us and with life science companies and SciAps with our community health system partnerships, we're looking at ways to develop tools that will do a better job of finding suitable trials for targeted patients and really expand the trial recruitment venue to community sites, therefore making it easier for both the oncologists as well as the patients to identify and enroll patients. So let's spend a moment on clinical complexity. First, a few stats. In the last decade, 88% more data points have been collected per trial. The amount of data that is now being generated for a single clinical trial is growing exponentially. Additionally, in meeting with clients, we hear quite frequently that it's taking upwards of 20, 25, 30 systems for them to actually do their work. And in order to get the entire process complete, upwards of 30 to 45 systems. And then lastly, there's been a tremendous growth in the number of actual procedures per trial, upwards of 70%. So how do we think about clinical complexity and helping our clients get treatments to market faster by overcoming clinical complexity? Firstly, talking about unified, right? As we continue to build out the Medidata platform, as we continue to work with sponsors, academia, CROs, with the clinical research community, try to understand ways that we can really help simplify and support new research paradigms. As Mark mentioned, basket trials, umbrella trials, seamless trials, pragmatic trials, they're all new paradigms for the industry. And as we work with clients, we continue to add functionality, some of which I'll talk about in a minute, to really help support these new research paradigms. We do a lot of work with NCI. I read an article, I believe it was last week, from NCI on their MATCH trial, which is one of a, I believe it has 40 arms in the study. So these new paradigms are actually critical for success in the industry and critical for us at Medidata to understand how we best support going forward. Flexibility. As we think about the amount of data and the types of data that are now being ingested for clinical trials, it's critical that the platform has the capability to both standardize, aggregate, and standardize data across the entire process, and we'll talk about that in a minute as well. And then lastly, a platform should be smart. It should be able to leverage the data that's in the platform to actually generate meaningful insights across the entire clinical process. So these are some of the ways that we think about overcoming clinical complexity, and let's talk about some of these in a little more detail. One of the other examples is the BEAT AML trial with the Lymphoma and Leukemia Society, which I believe is up to about 4 or 5 arms today, which in addition is recruiting additional arms as one of the premier ground-based and collaborative trials in the industry. So let's talk a little bit about the full Medidata platform and how we think about our capabilities supporting clinical complexity. So firstly, if we look to the right of this diagram, we see our RAVE family of products, which is around data capture and management. I'll come back to that in a second. Secondly, if you look to the right of this diagram, you see our trial planning and management suite of products, which is more the business side, if you will, of the process. And then within all of this is our enterprise data store, which is the underlying platform, as well as the analytics that we put and utilize throughout the platform in order to drive insights across the platform. So let's first look at the left side of the diagram, which is around data capture and management, the science, if you will. So firstly, if we think about onboarding the patient, as I discussed earlier, we talked about consenting the patient electronically. One of the other benefits of having a unified platform is that once that patient is actually enrolled on the platform, you then have the capability to use that data later on in the platform. So master data management, single source of truth. Once that patient is enrolled, they're then available for other systems like EDC, the randomization system, and ePro automatically without any additional setup. Additionally, as we talked about the sources of data now prevalent in precision medicine and specifically oncology, it's critically important that these data types such as images, genomics, digital biomarker data are all ingested along with the more traditional sources that we think about, such as in clinic or lab data. Mark mentioned imaging earlier in the hour, right? And if we think about how we ingest images today, many of the systems, in order to get images alongside the clinical data, this could take upwards of 7, 8, 9 systems sometimes. So that whole adjudication process of the images, along with the integration of the clinical data, is paramount and critically important to oncology research. And in the platform, we provide these capabilities to ingest all these different data types into a single platform so that you can then aggregate that data and have the ability to perform analytics on top of it. And then lastly, looking to the bottom left of the diagram around randomization and supply, once that data is combined onto the platform, it's then critical to have the ability to support these new paradigms, make mid-study changes in real time to support adaptive trials, support protocol amendments and other novel paradigms. So these randomization schemas must be adaptable in real time as the data are collected. And then lastly, thinking about things like coding the terms, we're using machine learning now to actually augment the coding process to really increase the probability of coding the terms quickly to really code terms over time in a fraction of the time. So looking at the right side of our portfolio around trial planning and management, this is the process side, if you will. We talked about, firstly, establishing a solid foundation for the trial, all the way back in trial design. Think about optimizing the protocol, minimizing the burden to the site and patients, then finding the best site. And then we also have the ability with our payments module to utilize the data to pay the sites quickly and fairly depending on when visits actually occur. And secondly, the study startup and oversight process. If we think about ClinOps and their role in starting up the studies and everything they have to do for site initiation and first patient in, that whole oversight process around study milestones, study tracking, building your ETMF throughout the process, that's all integrated into the platform. And then lastly, if we think about monitoring and CTMS, firstly from a CTMS perspective, the ability to have process optimization around that whole classic CTMS paradigm from study management to site monitoring, but then also as we link into risk-based monitoring and supporting new guidance from ICH, that ability to broaden the definition of, if you will, of monitoring from just being about a single visit and SDV to taking it to a full end-to-end approach to risk, thinking about building that rack all the way back in towards the study design, and then following your risk indicators throughout the whole process. So really going beyond that traditional data cleaning and monitoring to support that notion of risk throughout the entire trial process. So as we continue to build out the capabilities of the platform, one of the things we're finding is that the capabilities enable all roles to really work as one team on a single platform and to really excel. To have the ability in clinical data management to also pair that with machine learning to understand when there's anomalies in the data that perhaps the edit checks did not catch, and then be able to use the issue management and workflow to identify those issues and route them perhaps to a central monitor who then needs to take that information and perhaps send out a site monitor for a monitoring visit to check it. The unified platform really enables all of these roles to break down the traditional silos and work in new ways to really support and improve the aggressive timelines that are required in this space. And then lastly, just let me spend a moment or two on data abundance. A couple of quick stats here. There's been a 48% annual growth in healthcare data volume, the tsunami of data, if you will, that occurs in clinical research. Over 80% of the healthcare data today is unstructured, and there's 4 times the number of data sources per trial from 2017 to 2020. It's just a huge increase in the volumes of data that we've seen than we've seen in the past. And so if we think about overcoming the challenge of data abundance, the ability to collaboratively share data and use artificial intelligence and advanced analytics throughout the process is fundamentally changing the way clinical trials are performed. If we think about this enormous volume of data that is generated, whether it's billions of nucleotides or hundreds of laboratory tests or thousands of wearable sensor readings or millions of medical imaging pixels, if you will, it can present formidable challenges on how to structure, integrate, and interpret all of this data. And by having this unified platform, it really provides the ability to take all of that data and deliver fit-for-purpose, if you will, insights. And so I won't go into a lot of detail around our entire advanced analytics suite, but just let me hit on a couple of high points and a couple of the really novel things that we're doing in our data science area, specifically around oncology and for oncology research. So looking to the left hand of this slide, we've talked about a few of these, right? We've talked about optimizing trial design. by taking a look at protocol complexity and patient burden. We talked about study feasibility, also the ability to look at the sites that are in your trials to see performance, more the operational side of analytics. Additionally, operational efficiency, being able to look at various milestones, last patient, first patient to last patient, first patient in to last patient in, last patient, last visit to database log, and really understand the operational efficiency of your trial and how it compares. So these are the operational side of the analytics that we think about as part of our platform. And then looking on the right-hand side, if we take a look at things like central monitoring, which I just mentioned, to really use machine learning to detect when there's anomalies in the data, to really improve data quality throughout the process, across all the data sources, to identify ways that potentially would not be picked up from human intervention. And then two areas I want to spend just an additional moment on, with those being synthetic controls and our genomics capabilities that we're very excited about. So a moment on RAVE genomics. Today, ingesting and managing genomics data, as Mark mentioned, can be really formidable. It's a lot of data, it's time-consuming, it's error-prone. And by really improving and streamlining that process for every group that touches the genomics data is one of the key areas that we're focused on. So I believe Mark termed this more on the translational research side, but being able to subset patients, that whether you're looking at optimize target enrollment, whether you're looking at it to leverage specific biomarkers for protocol design, whether you're looking to integrate that data with other trial data, you'll have the ability to run the same quality checks. And then lastly, for biomarker discovery, advanced analytics, we have a tool for advanced analytics that really allows for exploration across the entire process. So one of the areas that we're very, very excited about is we continue to build out the capabilities to support oncology research. And lastly, just a moment on synthetic control arms. Given our 18 years in the industry and the amount of data that we do have the ability to start to look at ways in which we can use the historical trial data in novel ways. One of these ways is as a synthetic control arm or as synthetic control data. We have more information on this on our website, but we did about a year ago at ASCO last year published a study with a large pharmaceutical company that was actually looking at pushing the drug forward from phase 2 to phase 3, and wanted confirmation. And so by using the data to develop a synthetic control arm to make that go, no-go decision, that's one of the ways in which we're thinking about using this data. And clearly, it would be fantastic if we could use this data to actually be a synthetic control arm. Clearly, we must get the regulators on board in order to do that, and it'll probably be quite some time before it's accepted as definitive evidence. But we continue to work with our clients to think about how we can do that, why we think about other use cases like I just mentioned in order to progress the study from, say, a phase 2 to a phase 3, perhaps to augment a control arm if control patients have dropped out. Perhaps maybe there's a specific population that's needed to round out a patient profile. So again, this is one other area that we're very excited in the way that we're thinking about our data. We have a staff of data scientists that all they do day and night is work on the data to identify ways and develop insights to support oncology research. And then lastly, one more data point. You'll hear a lot more about this from us in the weeks and months ahead. But last week we did announce the acquisition of a company by the name of Shift, which is the world's leading platform for commercial and real-world data analytics. We're very excited about the possibility of closing the loop, if you will, between clinical and commercial, and really also, especially for the clinical paradigm, be able to use meaningful real-world data to support real-world evidence. So again, you'll be hearing a lot more from us on this in the weeks and months ahead. So with that, I know we only have 5 minutes, so thank you all for attending today. Thank you, Mark, for participating. And with that, let me turn it back to Sylvie to open it up for questions.
Great. Thank you, Michelle, for the wonderful presentation. Now, before we end the webinar, we would like to see if there is any questions from the audience. If you have any questions, please type into the Q&A box, and we'll collect your question now to answer. We'll pause for a while here now. So, thank you again for joining our webinar today. If you have any question in the future, or if you would like to know more about our solutions and the platform, or even a demo session, please fill out the survey that we are going to send you, email to you after this webinar. Our team will be in contact with you. Stay tuned for our upcoming webinars or other activities happening in the region, and hope to connect with you all again soon. Have a great day. Goodbye.